Good day, this is Professor Will, CBMD, PhD. Today, Saturday, March 23rd, 2019. It is 10:32:54 seconds Eastern Standard Time. And today I'm going to be doing a short study on agar. So let's get started here. Agar. Agar is a gel-like substance composed of a polysaccharide made by marine algae. Microbiology uses agar as a solid surface for growing bacteria and fungi because of its properties of melting and solidifying, holding moisture, and providing an inert surface for microbial growth. The gel-like characteristic of agar results from a polysaccharide called galactin, which is found in red algae. The red algae Galidium and Gracilaria serve as the main commercial sources of agar supplied to microbiology laboratories today, though they may be labeled simply as seaweed on the agar's container. Agar-based media enable microbiologists to grow a very wide variety of microorganisms in the laboratory for studying microbial behavior, enzyme activities, and morphology, cell or colony appearance. Agar's main attribute to its capacity to form a solid or a semi-solid surface. Solid agar enables microbiologists to perform the following studies: colony morphology of bacteria, yeast, and fungi, enzyme activity assays, antibiotic activity testing, and nutrient requirement tests. Semi-solid agar helps in studying motility and chemotaxis because motile species move through the semi-solid material while non-motile species will not move. Studies on nutrients, growth factors, and antibiotics have been aided by the ability of agar to permit certain substances to diffuse into it. Agar diffusion studies wherein an antibiotic diffuses into an agar medium previously inoculated with bacteria have been the main test method for assessing antibiotic activity in a test called the Kirby-Boya test. Purified agar granules disperse in the water when the mixture is heated to over 212 degrees Fahrenheit or 100 degrees Celsius. Then as the mixture cools to about 104 to 150 degrees Fahrenheit, 40 to 45 degrees Celsius, it solidifies in a translucent gel. Once gelled, it remains stable for several weeks up to 150 degrees Fahrenheit or 65 degrees Celsius. Solid growth media contains from 3.5 to 6% agar. Semi-solid formulas usually contain 0.5-1.5% agar. Types of agar. Nutrients are added to molten agar to make hundreds of varieties of microbiology media. When microbiologists refer to agar media, they mean the formulas containing water, agar, and all the nutrients needed by microorganisms for growth. Nutrient agars grow a diverse variety of bacteria or fungi. Specialized agars consist of formulas for growing only a certain type of microorganism. Selective agars and differential agars represent the two most commonly used specialized agars. Selective agars favor the growth of one type of microorganism over others. Saboro agar, for example, enables fungi to grow, but inhibits the growth of most bacteria. Differential agars give more specific growth responses and distinguish one type of bacteria from other types of bacteria. Differential agars do this by containing ingredients that enhance the growth of some bacteria while inhibiting or having no effect on others. For example, McConkey agar is a differential medium containing a red dye and the sugar lactose. It differentiates between lactose fermenting bacteria whose colonies turn red during incubation and non-fermenting bacteria which are colorless. Most selective and differential formulas take the form of agar media rather than liquid media referred to as broths. This is because the purpose of selection and differentiation is to isolate one type of microorganism from all others. By inoculating a solid agar surface in a technique called streaking, a microbiologist can produce isolated colonies on the agar. These single distinct colonies become visible during incubation. They are usually pure, meaning they contain the exact clones of a single parent cell. Pure cultures are necessary in many areas of microbiology, of which the most important are the following identification technique: selection of an antibiotic treatment, or carrying out of genetic engineering. None of these activities can take place without a pure colony of a single microorganism as their starting point. Techniques using agar media. Molten agar poured into either Petri dishes or test tubes takes the shape of the container once the agar solidifies, and these different forms of agar serve different uses in microbiology. In Petri dishes, molten agar spreads out into a flat, smooth surface that fills the dish. Once the agar has solidified, it takes the shape of the Petri dish, also called a Petri plate, and together they are known as an agar plate. A pour plate is any Petri dish that has had agar medium poured into it and allow it to solidify. The resulting smooth gel-like solid surface is then ready for inoculation. The terms pour plate and agar plate have equivalent meanings. Molten agar can also be poured into tubes instead of Petri dishes, tubes that are positioned at an angle. When the agar solidifies, allow the agar also to harden at an angle. This type of agar surface is called a slant. Slanted agars provide a larger surface area for bacteria or fungi that grows better in tubes than on plates. Molten agar poured into upright tubes, by contrast, constitutes an agar deep, also called an agar butt. Deep agar tubes are useful for studying bacteria motility in semi-solid medium. Microbiologists inoculate agar plates by one of two methods, streaking or spreading. Streaking is done with a small wire loop that holds a drop of broth culture, and by dragging or streaking, the loop is drawn gently over the agar surface. By doing this, a single line of colonies forms when the plate incubates. The colonies at the outermost end of the streak are often isolated from the others, and these isolated colonies are considered pure colonies. Spread plates differ from streak plates by holding an inoculum made as a wide swath across the agar surface. After incubation, a spread plate contains a uniform layer of adjacent colonies called a lawn. The properties of agar have made it superior to other gel-like substances for procedures in microbiology. Agar has only two disadvantages. The supply of agar worldwide varies depending on seaweed supply, and because agar is a natural material, it varies in composition from one batch to the next. Some compounds serve as agar substitutes: silica gel, carrageenan, gellan, gum, and pectins. All of these, except silica gel, contain long polysaccharides made by microorganisms. Because these materials have natural sources, they vary from batch to batch, on occasion in the same manner as agar. Silica gel requires an extra step when being used in microbiology because it contains silicon dioxide SiO2 units and contains no carbon. To use silica gel for growing bacteria, a microbiologist must add a carbon source. Despite the greater availability of silica gel, carrageenan, gellan gum, and pectins as agar substitutes, microbiology laboratories prefer the properties of agar over all other substances. Today's is Professor Will, CBMD, PhD, today Saturday, March 23, 2019. This concludes my brief description on agar. Hope you enjoyed listening. Have a great day.